article's title is Phytochemical, Antioxidant, and Antitumor Studies of Coumarins Extracted from Granny Smith Apple Seeds. Recent trends in drug development have refocused on the isolation and identification of natural products from natural sources, mainly due to their long history of safety and structural diversity. The idea behind this work went with the utilization of food processing waste and byproducts as innovative ingredients due to their considerable content of natural compounds. Apple seeds are an example of such processing byproducts. They contain a wide variety of natural compounds with different bioactivities that may have a potential use as a functional ingredients in the nutraceutical and pharmaceutical preparation. Why Granny Smith apple seeds? According to our survey, which has been performed on September 2019, a number of research papers were found studied the phytochemicals of the peel, pomace, or the fruit flesh of this cultivar. But our attempt to find studies recording the chemical composition or the bioactivities of Granny Smith apple seeds have been failed. This has been inspired the teamwork start with this study. As we see in the picture below, the Granny Smith apple is distinguished by its light green flaked peel with white, juicy, and crispy flesh. Australia is the birthplace of the Granny Smith apple. It was found by Maria Smith in the 19th century. This is why it is called the Granny Smith apple. Our teamwork is focusing on the study of coumarins in terms of their bioactivities, chemical synthesis, and isolation from natural sources because these compounds are a large and structurally diverse family of substances which exhibit a multitude of bioactivities. Coumarins are characterized by their low molecular weight, easy synthesis, high bioavailability, and limited toxicity. As we see here, this is the chemical structure of the coumarin scaffold. It is defined as an oxyheterocycle of a two-edged chromium 2-on. This work aimed to isolate the coumarins found in the seeds of the Granny Smith apple and to explore their in vitro antioxidants and antitumor activities. This aim is achieved by satisfying the following objectives: extracting the apple seeds powder by utilizing 4 different solvents and 3 styles, submitting the resultant extracts to a qualitative screening assay, isolating and purifying the natural coumarins, characterizing the chemical structure of the purified coumarins, and finally studying the in vitro antioxidant and antitumor activities of the isolated coumarins. Material and methods. A Granny Smith apple purchased from the local market and identified by scientific committee at the Agriculture and Forestry College at the University of Mosul. The fruits sliced manually and the acquired seeds were dried in shade at room temperature for 2 weeks. Crushed and seeds providing fine powder. This seed powder was extracted with 4 solvents: water, methanol, chloroform, and normal hexane. 3 methods were applied. The first technique was kinetic maceration with a mixture of the seed powder and extracting solvent, macerated using shaker water bath for 3 days at 30 degrees centigrade. The second method was ultrasound facilitating extraction. The mixture of the seed powder and the extracting solvent sonicated for 30 minutes at 30 degrees centigrade. And the third technique was microwave facilitated extraction. The mixture extracted for 5 minutes. With each technique, extraction was performed in 3 styles, including non-serial, serial descending, and serially ascending style according to the polarity of the solvents. In the non-serial style, the seed powder extracted with every single solvent separately, while in the serial style, the powder was extracted with the first solvent in the order. The extract mixture was filtered and the filtrate subjected to phytochemical analysis, while the residue was extracted with the solvent next to the first in the applied order. These events were also applied for the third and fourth solvent in the same manner. In total, 36 extracts were obtained, all of them subjected to the phytochemical analysis in order to choose one of them for the isolation of coumarins, as we are going to see in the next slides. Isolation of coumarins. The chloroform extract of seeds powder acquired from the ultrasound facilitated extraction, which operated in a non-serial style, was concentrated to dryness and reduced pressure. The presence of lactone is a key structural motif for of coumarins, which are hydrolyzed once attacked by a strong nucleophile like NaOH into water-soluble salts of cis-dynamic acid derivatives. The acidification of these salts results in the restoration of the original coumarins. This is the chemical scheme was applied as an isolation method for coumarins in this study. Ascending CLC technique was used to detect the number of coumarins present in the extract. The separation of coumarins was carried out by column chromatography. 5 simple coumarins named R1 to R5 were detected. Each one revealed a single spot in CLC in different element systems. In vitro biological studies, antioxidant capacity of the isolated natural coumarins was detected by testing their efficiency to scavenge GPPH and hydroxyl radicals, utilizing vitamin C as a reference compound. The antitumor capacity of the isolated coumarins tested using MCT dye and 2 cancer cell lines, including HeLa and MCF7, while 5-fluorouracil used as a reference. Results and discussion. The resultant 36 extracts were examined for the presence of certain primary and secondary phytochemicals. The results indicated that coumarins were found in chloroform and methanol extracts, which obtained from the applied extraction techniques and modes. The chloroform extract from the non-serial ultrasound facilitated extraction was selected to isolate its coumarin ingredient. The selection was based on the absence of certain phytochemicals in this extract, including flavonoids, alkaloids, tannins, and fixed oil, where their presence may interfere with the isolation of coumarins. Interpretation of the chemical structures. The chemical structures of the isolated coumarins were elucidated depending on the chemical evidence obtained from the consensus and on the analysis of their SCIR, proton NMR, and carbon-13 NMR spectra. This elucidation revealed that the isolated coumarins are novel with the exception of the compound R1. It was found that there is a remarkable agreement between the physical properties and the spectral data of R1 and those of oxygenol. In this figure, we can see the chemical structure of the isolated coumarins from the Granny Smith apple seeds, including the R1 and the remaining 4 novel coumarins. Regarding the in vitro biological studies, this figure represents the results acquired from the anti-radical activity of the reference and the isolated coumarins. We can summarize the results of the anti-radical activity of the isolated coumarins in this figure. The R3 compound with its 2-phenolic hydroxyl group was showed the best scavenging capacity of the GPPH and hydroxyl free radicals, while the compound R5 with the strongest electron donating group was showed the second best anti-radical activity. On the other hand, the compound R1 with its only 1 phenolic hydroxyl group was showed the least activity in the scavenging of the free radicals. Regarding the anti-tumor activity, we can see again that compounds R3 was showed the best anti-tumor activity against HeLa, MCF7 cancer cell line, followed by the compound R5. In many in vitro and in vivo studies showed that there is a considerable connection between the oxidative stress and the propagation of cancer, and the intake of exogenous antioxidants can limit the free radicals damage to essential cellular components that can reduce the risk of the development of cancer, propagation of the cancer, or both. In conclusion, this work demonstrated a success in the isolation and structural characterization of novel simple coumarins from Granny-Smith apple seed. The isolated product showed promising antioxidant and anti-tumor activities with a positive correlation between them. It's proposed that the anti-tumor activity with the isolated coumarins can be attributed to their antioxidant capacity, and this may serve better in the design of new antioxidant and anti-tumor agents. Thank you for your listening. If there is any question, you can find me on this email.
Thank you so much. Very nicely presented and very you concised well within the time. The presenter has shared her email address. If anyone has any question, you can always collaborate and communicate.